This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 382, recorded on March 25th, 2016. Hi, everybody. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today here in the Microbe TV studio, Dickson Despommier.
Hello, Vincent.
I don't even need to ask how you are, because you've been pretty well today and yesterday.
Feel great.
Feeling great, excellent.
Yep, feel
and
great.
What's the weather like here?
You know, it's actually gotten better since this morning, and it started out as a muggy-looking, kind of a rainy-ish, misty, yucco day, and it's morphed into a partly cloudy, in the high 50s, maybe even low 60s, less humid type of afternoon. Makes me want to go outside and
Go ahead.
stand waist deep in a trout stream someplace.
You can go right now.
No, I could never miss this show.
It's 18 Celsius. You know, last night when I left, it had gone down to 6 Celsius.
Yeah.
It got nippy.
It was a
Interesting
little coolish.
weather. Also joining us from southeastern Michigan, Kathy Spindler.
Hi, everybody. Here it is 1 degree Celsius,
Ooh,
Oh, my
35 degrees Fahrenheit.
what an interesting
What
pattern.
happened to all your
Bright
tulips?
and, bright and sunny, blue sky, puffy clouds. It's supposed to get up somewhere around the 40s.
Did it freeze overnight?
It was just below, it was 28 when I got up.
So all the flowers, did they get hurt by that?
No, a lot of them aren't blooming yet, but even if
Ah.
that kind of freeze wouldn't hurt the bulbs
Mm.
that are blooming at this point.
Also joining us from western Massachusetts, Alan Dove.
Good to be here.
What do you got there in the
Cloudy, rainy, fog, 5 Fahrenheit.
Got what we had this
Yuck.
morning.
Oh, interesting. Well,
Yes.
Dickson, would you be interested to know what the weather's like in Utah?
I'd love to
We
know.
could find out.
Really? How
because-
would we ever do
Well,
that?
I'll tell you, by the magic of Skype,
I can't believe this.
we have 2 guests, and one of them really needs no introduction because he is on one of the other podcasts in the Microbe TV empire, as Carl Zimmer calls it. He's at the University of Utah. Nels and Ede, welcome.
Hey, great to be here and in the company of science podcasting friends and mentors. I'm excited.
You have been, how many times on TWiV before? Once or twice, I don't remember.
I have been on TWiV once before. So this was at the ASM meeting in Denver
Right.
a few years back.
That's right.
Yep,
And
we did
Kathy was on that one too, right?
Kathy was on it. We did it in vivo, as you,
Right?
as you guys say.
And, you know, Nels, we had you and Tom Schenk.
Yeah.
And I remember when
Good
we
company.
started talking to Schenk, you whipped out your phone and started checking your email.
No, I was checking. You know what I was doing, Vincent, was the Fahrenheit to Celsius conversion, so that I
Okay.
wouldn't be caught flat-footed.
Ah.
So here, and to do that now in Salt Lake, we are looking at 6 Celsius under partially cloudy skies. A beautiful day with sun peeking in and out across the snow-capped mountains.
The Wasatch.
Wasatch out to the Okers.
Yep. And how has been the level of the Great Salt Lake? Has that gone up or down since you've last?
Yeah, it's been pretty low,
low.
almost on historically low levels. It
Interesting.
will now refill, at least temporarily, this spring as the snowmelt starts to come down.
Right.
But its surface area can vary dramatically.
Yeah.
Yeah. Yep.
So today we're doing a little podcast cross promotion. So Nels, of
Crossover
course,
episode.
crossover episode. Yeah,
Hybrid.
this is. We could have done this. So Nels, of course, is my co-host on TWiV, This Week in Evolution. If you don't know about it, you should check it out at microbe.tv
Should rename it This Era
TWiV.
in Evolution or something. Come on.
Yeah,
And now-
I do have some friends who noticed like that this week in evolution, but
It's a little short time for.
it's a little short time frame. But I'd like to double down and say we could even talk about this day in evolution when it comes, especially to these microbes, these viruses.
Yes.
There you go.
Things move pretty fast.
They do.
So Nels, I suggested to Nels that we do this paper on TWiV, and he said, "Nah, it's not really good to do my own papers on my own show." So,
Sure.
but I wanted to do it because it's a very cool paper, as you will see. So we're going to do that here today. And we're very fortunate to have one of the 3 co-authors of the paper joining us. He is a Jane Coffin Childs postdoctoral fellow over in the University of Utah. Ed Chuong, welcome to TWiV.
Hi, happy to be here. First time on TWiV, not like Nels, but I guess he's taking me under his wing.
So you are a joint postdoc with Nels and Cedric Fichot, right?
Yes.
Is it correct to say a joint postdoc?
We actually, their labs are adjacent to each other, and I guess I occupied the estuary intermediate zone between them. So it's truly both in physical
the ecotone
and an intellectual.
The two.
I was going to say, Dickson, is that an ecotone?
It certainly is.
So Nels, of course, needs no introduction because everyone knows his history from being on TWiV and also TWiVo, but I'd like to find out where you're from, Ed, and where you were educated. Where are you from originally?
Oh, sure. I was born in New York, moved to California at a very young age.
What a great state to be born in. Where, in New York City?
Yeah, New York City.
Wow, Manhattan, Queens, Bronx, Brooklyn?
Manhattan. I forget what the hospital name is. Cornell Hospital?
Yeah, New York.
I was only there for about a month, so
New York.
I didn't gain consciousness.
New York Hospital, yeah.
Yeah.
Although now it's Weill Cornell Hospital, right, Dickson?
Yeah.
I think it's changed. Yeah. So you weren't here for very long in the city, huh?
No. So I mostly grew up in Southern California, in Irvine.
Irvine, wow.
And then in Irvine, so it's sort of between Los Angeles and San Diego.
We have good friends in
Irvine is.
We have good friends at the university, and people like Bert Semler and Hung Fan and Luis Villarreal.
Roz Sandry Golden.
Yeah. Erwin Sherman. I knew Erwin Sherman.
Who's Erwin Sherman?
He's a parasitologist that trained with Bill Traeger at Rockefeller before he went out to take a job at Irvine.
So you grew up in Irvine, Ed?
Yeah. Went to high school there, just 5 minutes away from UCI.
Right.
And then for undergrad, I went to UC San Diego, so I moved an hour south. And that's where I studied. They had a new major in bioinformatics, so that's what I studied there.
Oh.
So I got a little bit of lab experience there. I worked with Dr. Hopi Hoekstra, who's an evolutionary biologist.
Oh, wow.
There.
That's before she went to Harvard.
Yeah, her early years. Yeah, so I was like her first undergrad. So
we want to get her on TWiV, right, Nels?
Yep, we're working on it. She'll come.
Maybe Ed is an in, right?
Yeah, exactly.
So I did undergrad there. With Hopi, started getting interested in evolutionary biology. Had a little project on the placenta, studying rapidly evolving proteins in mouse placentas, so that was interesting. And then for graduate school, I went to Stanford, and I was in the Department of Genetics there, and I worked with Dr. Julie Baker, and she also studies the placenta, so I just couldn't get enough. And there we were studying more like the evo-devo evolutionary development of the placenta. It's a very interesting organ. It evolved fairly recently in mammalian evolution. So we were just interested in those kind of basic questions. But then I guess in the course of the thesis project, I somehow landed in endogenous retrovirus land. So I think I've become hooked ever since into the virus world.
Well, isn't it true that we owe the placenta in part to endogenous retroviruses?
Exactly. So the placenta is just a super, even more so than many other organs, seems to have an intimate relationship with viruses in one way or the other. It could be that viruses like going through the placenta, but as of course, the placenta seems to be reliant on a domesticated and endogenous retrovirus for its development. So that was kind of the line of research that I followed. Decided I really wanted to get more into, thinking about retroviruses and evolution. So that's what led me to Utah to join the labs of Nels, Elde, and Cedric Fichot.
So when you applied for a postdoc, did you apply to both of them or one? How did that work?
Actually, it was Hopi who kind of just encouraged me to come out to Salt Lake and visit. And it was initially to interview with another professor, Gab Carden, who's also in the department. But then I was just looking through the website, and I saw Nels was here. I'd seen him from his interview circuit when he was on the job market. I first saw him at Stanford, and I knew Cedric from his his many reviews about endogenous retroviruses. So seeing the two of them, I just couldn't pass up the opportunity to visit their labs. And
how long have you been there now?
2.5 years.
Okay.
Yeah, 2.5 years.
And just to maybe add on the other side, when Ed came by, there's sort of this confluence of Cedric Fischott relocated his lab from UT Arlington to Utah. And then not long after that, Ed came and visited. And for both Cedric and me, it was super clear that this guy, if we could get him to come, it would be amazing. So we were super stoked when he said, he was going to come and do his postdoc.
Nice. Well, the paper we're going to talk about today is pretty amazing. So you guys, you lucked out all around, all three of you. That's great.
Agreed.
And before we do that, I want to tell everyone that this episode of TWiV is sponsored by the Agar Art Contest of the American Society for Microbiology. It's the second one. They did one last year. Very successful. Lots of entries. And what you do here is you get a plate of agar. It can be any kind of agar you'd like. As we were talking about yesterday on TWiP, it could be blood agar, or it could be chocolate. It could be McConkey's or just plain old LB agar, whatever you want. And you put your favorite organisms on in some artistic way. They can produce colors, so you can use Serratia marcescens, for example, which would be red, etc. And make an piece of art, a work of art, photograph it. We don't want the actual plate, but we would like a photo of it. And you send it in, and it'll be judged. And I am on the judging committee once again. And this is a lot of fun to look at all these things. So all the instructions are at bit.ly/agarart2016. The winners and some runners-up will be showcased at the big ASM microbe meeting in Boston this June. And, your photos will be put on display somewhere. And then the winners will also get a free ASM book of their choosing, which undoubtedly should be Principles of Virology, but, you may not have that good taste. Who knows?
You may already own it.
They may already.
And it's a great contest because, art has been part of our culture for so long, and now our culture can be part of art.
Ooh.
No kidding.
You are awesome. Oh, the awesome Alan. Wow. So the guidelines again are at bit.ly/agarart2016. And Alan and I will both be at ASM Microbe.
Yes.
So we will come by and see the winning works of art. It's pretty cool. Very nice stuff. All right, so we move to your paper, which was... Oh, so you guys know that... of course, you know that Carl Zimmer wrote a Times piece on your article last week, and he also made it his pick on TWiV last week when he was here.
Nice.
Oh, no way.
Yeah.
Way.
Did you know that, Nels?
I did, and I was super excited because, well, first of all, I'm a huge fan of Carl Zimmer, and so it was really fun to have that exposure and to get Zimmered, as the verb is sometimes used these days.
Zimmered, huh?
Uh-huh.
Zimmer bump.
Yeah, exactly. Yeah.
Wow. So speaking of bumps, Zimmer just hit 250,000 followers on Twitter. And one of TWiV fans say, "Hey, you got the TWiV bump."
There you go. You put him over the top, Vincent.
There you go.
Very generous of you.
Speaking of which, though, last week we were talking about Neil deGrasse Tyson and how many followers he had. He has 5 million.
I'm so jealous.
Out. Yeah.
I am so jealous.
More exposure.
And he only follows 47.
Yeah.
Isn't that interesting?
Yeah. About 5 of them are also astrophysicists, and 1 is Carl Zimmer.
Nice.
So you went to see him this week, right?
Yeah, he did an evening performance here, and it was great. It was almost 3 hours long. He did a little bit of Q&A at the end, and he has evidently 15 different presentations that whoever the presenters or whatever could have chosen from. And so he gave one to us that was about sort of the cosmic view of things, and totally entertaining. He came out immediately, took off his shoes, and he did the rest of the evening in his sock feet. And he walked off stage carrying his shoes when he was done.
What's the point of that? Do you know?
I just wanted to be comfortable, I guess. I don't know. And then he did a couple ballet type
kind of a
movements.
Mr. Rogers entry.
Sort of, yeah.
Does he have visuals, I guess, right?
Yeah. He had a PowerPoint presentation. One of the cool things he showed was the periodic table, and he can color all the elements however he wants. And so the second view of it, he had it colored by the flag of the country that had discovered the element.
Oh, wow.
And had that tallied up, and that was really cool. There were about 7 or 8 that were known since ancient times. So that had a different coloration. But
Nice.
the UK was first and the United States was second. And somebody in the audience yelled out, "Hey, Canada!" because they had one.
So how many people were there?
The auditorium was full, so it's over 3,000 capacity.
Goodness.
Just like when we do a TWiV, you know. So he's really an actor, isn't he?
Just total entertainer.
Yeah.
He was passionate about the fact that, part of his points were, how the U.S. is falling behind in science because it's not making investments in science, the kinds of things that need to be done. So, yeah, I don't know that he's acting. I think he's performing. He's just telling what he thinks.
I'm sure. No, I meant performing because he clearly does that.
Yeah.
I was asked to go on his show a couple of weeks ago. He wasn't going to be there, but that would have been cool. Maybe I could have gotten some more Twitter followers.
I'm sure. Yeah.
Anyway, cool. That's cool. Well, we have to morph TWiV into something. We could have all 5 of us, take our shoes off and
That's right.
do things. I don't know if
Walk
anyone.
across a crosswalk.
I don't know if anyone
Barefoot
would.
virologist.
That would be a good title, the Barefoot Virologist. Yeah,
Wow.
good idea. All right. Let's get on to it's Ed's paper. It was published in Science last week, and Ed is the first author, and Nels is the second, and Cedric is the third. And Nels and Cedric have the little dagger thingies. I don't know what you call those.
Right.
So usually the first and second author, you might say these two contributed equally, but this time we have 2 PIs who contributed
equally.
Very interesting. It's regulatory evolution of innate immunity through co-option of endogenous retroviruses. So Ed, can you tell us how you came to do this piece of work?
Yeah. So when I started my postdoc, we're planning what I would be doing during my postdoc. And so at that time, which was, maybe around 2013, I think one of the really, exploding in my mind, I guess topics or issues in the field was the idea that transposable elements, including, endogenous retroviruses, basically these mobile elements in our genome, they're very abundant. And it was starting to become clear that they may have some regulatory activity in the genome. So people were starting to use technologies like ChIP-seq and basically functional genomic approaches to assay what regions of the genome might be functional or regulatory transcribed. And a lot of these studies, a lot of them coming not from transposable element or virus labs, sort of all independently converged on this observation that these endogenous retroviruses in our genome are not just sitting dormantly, but seem to have really cell type or tissue specific regulatory activity. And the implication there is that these elements could then be a very potent force in driving, species evolution, adaptation of different biological processes. But that was kind of the state of the field in that we were able to see this regulatory activity, but then to actually functionally go in and know if this activity was actually doing anything important for the host, or if it was just kind of a remnant parasitic activity of the virus, that question was still unknown. And so I think that was kind of our motivating question for this project.
So the first couple of experiments are in fact pure bioinformatic analysis of existing data, right?
Yes.
And the first one being ChIP-seq data. Why don't you tell us how that works?
Yeah, would you like me to explain ChIP-seq a little bit first?
Yeah,
please.
Please do it, especially for Dickson.
Yeah, sure.
Only for
Well,
Yeah,
and for
so-
Caleb, the others in that.
Sure. Come on.
Okay, yes. This is kind of where my background comes in. So I'm not, I'm sort of starting in virology with this paper, but my background is really in regulatory genomics, functional genomics. So one of the techniques that's come out, relatively recently and now really kind of a mainstay in a lot of these kinds of labs is ChIP-seq, and it's short for chromatin immunoprecipitation followed by high throughput sequencing. Essentially what this does is it lets you create a map of all the places where a certain transcription factor binds in the genome. And so here we're interested in the interferon response, specifically the interferon gamma response. And in this response, which is, turns on during infection, at the regulatory level, it's controlled by a transcription factor named STAT1 or IRF1. These are two different transcription factors that are involved in basically turning on genes known as interferon-stimulated genes, which serve as our first line of innate immune defense for the cell. So basically, these transcription factors work by moving into the nucleus and then binding to specific regions in the genome that are nearby these interferon-stimulated genes, and that binding kind of turns those genes on. So the first question is, okay, where are these transcription factors binding? So now, for a while, to get that kind of map on a genome-wide scale would be unheard of, but now with ChIP-seq data, we can get that data relatively straightforwardly. So in this case, I was just using published ChIP-seq data sets that were produced by other labs, also other consortia such as ENCODE. So we're kind of like a research parasite. We just downloaded
parasite. I was just going to say.
Nice.
Yeah. So we just started by downloading this data from these resources and basically, remapped and reprocessed this data using our own pipeline. So all this stuff was consistently analyzed. And so now in our hands, we had these maps of where STAT1 and IRF1 were binding in a couple of different cell types just to get kind of a broader survey. So we looked at HeLa cells, K562 cells. These are both cancer cell lines. And we also looked at a primary CD14 positive macrophages from human. So we had these maps, and our first question was basically, well, we have all these binding sites, and in some cases tens of thousands of binding sites. How many of these land in transposable elements, or what's the contribution of these elements to these binding sites? So that analysis, you can basically just overlay the two. You can overlay the map of where these transposable elements are. You can overlay the map of where these binding sites are and look for that intersection. And the short answer is we found a lot. We found that, consistent with previous studies that have done similar kinds of analyses, anywhere from 10 to 30% of these binding sites come from these transposable elements. So that was pretty interesting to see.
And what makes up these transposable element sequences?
Yeah, so we did, I won't go into the details here, but we did some, sort of deeper analysis to see, are there any transposable element families or types that are overrepresented in this set? Because, 50% of the human genome, at least, is transposable element derived. So it's really no surprise that some binding sites are going to land in transposable elements. But one aspect that would have been exciting is if, and this goes back to the, the early hypotheses of Barbara McClintock and so on, but these transposable elements, they can potentially carry, sequence patterns that bind to STAT1 or IRF1. And if they have that, and if they amplify throughout the genome, they would be, in essence, copying and pasting that sequence throughout the genome. And so we looked for instances of that, where we would see, one kind of transposable element bound by these transcription factors more often than expected by chance. And we calculated chance by just shuffling these peaks throughout the genome a couple thousand times to get kind of a background expectation. And what we found there is we found 27 families of transposable elements that seem to be enriched for these binding sites. So these seem to, contribute more than, their fair share, I guess, of binding sites in the genome. And what, surprised us was that the majority of these families were actually endogenous retroviruses that were largely primate specific. So that's what really got us onto this endogenous virus line.
So basically, it's a series of analyses where you're looking to see. The null hypothesis would be that these transposable elements just randomly distributed themselves around the genome, and you're looking and seeing if there's in fact a non-random distribution that favors these particular types of sites, and that is in fact what you see.
Yes, exactly.
Yeah, and from an evolutionary standpoint, you can kind of think about it a little bit the other way around. So the process of the dispersal is still random, but then how selection acts on it is not so random.
Right.
Yep.
I'm absolutely struck dumb by the alignment of the years and how you got this graded series all the way up to the present when you shuffled them to show how they related to each other. That's spectacular.
Yeah, this was a pretty striking.
What did you drink when you found that out? What year champagne did you crack open? Really, because that was just spit out by a computer, I presume. And you're sitting there waiting for these results, and all of a sudden it just shows this linear. So this is fabulous.
That's how a lot of work is done these days, Dickson.
But it's fabulous anyway. A lot of work done today is not fabulous. This is fabulous.
Are you a little jealous, Dickson?
Not at all. No, I'm absolutely stunned and pleased to see such a parsimony of evolution.
Well, there are some experiments here too, Dickson. Although bioinformatics is an experiment in itself, right? Ed, you would argue that you don't have to mix small amounts of liquids to do an experiment only, right?
No, but I think bioinformatics is a really powerful way to identify, really interesting experiments that other people may not have come upon.
Vincent, you mix the liquids after you get the results.
Okay, I've been wrong all these years. So the next experiment, Ed, is also a bioinformatic experiment, right?
Mm-hmm. And just to answer Dickson's question, so we drink whiskey out here in
yes.
the foothills of the Wasatch Mountains.
There you go. Absolutely, absolutely.
Single malt, blended.
Mostly rye blends. Those are the fashionable ones. There's a great distillery called High West in Park City that's doing some interesting stuff with rye whiskeys.
Right.
Okay. All right. Now back to the science. Although one could argue that rye blending is a science.
But it's all from nutrition.
What's the next bioinformatic experiment, Ed?
Oh, we're talking about the expression one.
Yeah.
Mm-hmm. Yeah. Okay. So part A was saying, okay, without even thinking about the genes themselves, do we see a lot of these STAT1 binding sites coming from endogenous retroviruses? Which we do. And so the next question is, if these, ERV, and I'm going to say ERV for endogenous retrovirus sometimes it's just the way I've been brought up to say it. Sure. But if these ERV-derived binding sites actually, I guess the next question is whether these ERV-derived binding sites might actually contribute to the interferon, gene regulatory program. Mm-hmm. And so basically what we did is we took all of the viral elements that were bound by STAT1 or IRF1 based on the primary macrophage data. So just looking at that data set. And there were about 7,000 of those ERVs. And then we basically asked, what's their distribution relative to the next closest interferon-stimulated gene? And so the interferon-stimulated genes, we separately determined those. We found about 2,000 of those based on an RNA-seq data set of interferon gamma-stimulated CD14 cells. So this is a separate data set, but produced by the same lab. So we're kind of treating them as a matched data set. And basically what we found is that a very striking enrichment of these ERVs near these ISGs or interferon-stimulated genes. So for instance, in that figure, we're seeing that 300 of these ERVs, of these STAT1 or IRF1-bound ERVs, are actually within just 10 kilobases of a significantly interferon-stimulated gene, whereas if you were to just look at all other ERVs kind of by chance, you'd only expect about 74. So this is a very, this is more than a threefold enrichment. This is very striking. And then as you can, if you were looking at the figure, you would see that if you were to look at a farther distance, like say 500 kb away, where you're not really expecting it to have an effect, then these STAT1 bounders kind of look like the background expectation. So this kind of gives us a pretty clear signal that at the very least, these viral elements that are bound by STAT1 are definitely right next to, a lot of them are right next to the interferon-stimulated genes. So that gives us kind of this correlative hint that these may be actually responsible for, at least are contributing to the regulation of these genes.
You're essentially quantifying the amount of selective pressure that was likely put on these spots with these genes.
That's the hope. That's one implication,
right.
But I think
there are other ways you could get that outcome, but that's the presumption is that the most likely, most parsimonious explanation is that there was some selective pressure that led to this distribution.
Yeah, or absence of selection that perhaps these are not bad for the host. Yeah. Because a lot of these TEs, I think the vast majorities are persisting simply as neutral mutations and probably have pretty subtle effects. But these 300 may have big effects. I think that's what we're thinking. So
next, you focused in on a particular endogenous retrovirus called MER41. Tell us how you got in on that.
Yeah, so that one's super interesting. Yeah, so we're continuing to zoom in. What we're interested in now is looking at one specific family. And so we chose MER41 for a number of reasons. One is that it was the most, by far the most enriched for STAT1 binding sites across all of our cell types. So this is saying, it's not a cell type specific or not quite so cell type specific of an ERV. And it had actually also been previously identified in a PLOS One paper in, I believe, 2011 or 2010. And they were just doing a bioinformatic analysis of the same data set, actually. But they actually identified MER41 as a sequence artifact. When they were looking for overrepresented motifs in STAT1 binding sites, they were finding bits and pieces of MER41 that didn't even have anything to do with STAT1 binding. But they were kind of saying, well, look, when you're doing these kind of ChIP-seq analyses, you want to consider repeats and potentially ignore them so they don't, mess with your analysis. So here we're actually taking a different look, and we're doing the opposite of ignoring on it. We're focusing on it. And so with MER41, kind of the first thing we did as a check was we took advantage. So one thing is that these MER41s are very strongly enriched in STAT1 binding sites. But one of the things we're starting to learn in the regulatory genomics field is that the binding of transcription factors alone is actually a very pervasive phenomenon throughout the genome. So you have a lot of sort of solo binding sites of different transcription factors, and really other signatures kind of can help in terms of determining whether something actually is acting with functional regulatory activity. And so one of these signatures is localized enrichment of a histone modification known as histone 3 lysine 27 acetylation, or H3K27AC. And basically, with known functional enhancers, you often see co-localization of both transcription factors and these enhancer-associated histone marks at the same place. And that really helps drive in, or really helps make a more robust prediction of whether that's a functional enhancer. So basically what we're showing here is, or what we found is that with MER41, not only does it gain STAT1 binding upon interferon treatment, it also gains very clear and strong H3K27 acetylation enrichment. So that, if you were to show that kind of data to someone who just studies enhancers, they would be, they'd say, yeah, it's an enhancer, even if you didn't tell them it wasn't a viral element. So it looks like an enhancer by any other means, I guess, is the point. And we were then able to show that we zoomed in even closer on the sequence, and we found that where that STAT1 binding site was occurring based on ChIP-seq, basically perfectly coincided with a tandem pair of sequence motifs that are known to recruit STAT1. And so this is called the gamma activated site, or GAS motif. It's TTC followed by 3 any base pairs followed by GAA. And this is a known site that's been known to recruit STAT1. And what we found is that this MER41 element has 2 of them right next to each other, right in the middle of the element.
Yeah, that's pretty neat. And the histone H3 lysine 27, that's also figured out by ChIP as well, using an antibody to that modification. Is that correct?
Yes, exactly. So the data set that we parasitized.
Yeah. That's pretty cool. Parasitized. Yeah, so we're still parasitized.
Commensalizing it.
We also call this dumpster diving.
Really?
Yeah.
That's good.
Oh, that's cool. Dumpster dive.
We're learning from the genome. We're learning from evolution.
You can only do that for junk DNA, though, can't you?
Yeah, you're raising an interesting controversy.
I'll bet. Yeah, there is no such thing as junk DNA, right?
I mean, most other people ignore these elements, so it kind of is it is to our advantage.
Before we leave this, I want to ask about the fact that you found a lot of these ERV-like elements as in your results, but you also found some lines and signs, and I can understand those. But you also point out some that are just labeled as DNA, and the terseness of a science paper doesn't allow me to figure out what do you mean by those DNA elements, like MER107 and TIGR7.
Yeah, I mean, so it's not like a exact class. It's like a hodgepodge of different families of different types of transposons. So if we think about transposable elements, you can really broadly divide them into, basically DNA transposons, which basically, don't really go through an RNA intermediate when they sort of move across the genome, replicate or move, whereas ERVs and lines and signs, those are known as retrotransposons. And so in their life cycle, they get transcribed, and then that transcript then gets reverse integrated. So that's the difference.
Okay. Thanks.
So, Ed, if you did this analysis with any transcription factor that binds DNA, because here you did STAT1 and IRF1, if you used IRF3 or 7 or something else, would you also get localization within transposable elements, or is this limited to certain types?
Yeah, it's a great question. I think, you would probably pretty much see the same thing for the majority of transcription factors. And that's, not surprising now, but was surprising when that was first discovered. And, I think cells such as embryonic stem cells, placenta stem cells, tissues involved in early development, those have actually primarily been the focus in this field. And so transposable elements, including endogenous retroviruses, have been well documented to show prolific regulatory activity in those cell types. A more recent study by Ting Wang's group, where they looked at all the ENCODE cell lines and all the ENCODE TFs, they found that pretty much any cell line, any TF you look at, a good percentage of those binding sites, from anywhere from 5 to 30%, you can guess will probably come from transposable elements. So I think in terms of that, this doesn't seem to be too specific to the immune system or the STAT1 regulatory network. But I think a very comprehensive analysis has not yet been done.
So that's what I was trying to get at. Like, roughly what percent of all the transcription factor binding site have been introduced by mobile elements? You said up to 30%. That's really remarkable, right?
Yeah, I think Ting Wang's thing was 5 to 40%.
Wow. And
the number tends to be lower in somatic tissues, such as like the fibroblast. I think my hunch is that immune cells might have a little bit more than other somatic tissues, as we'll get to. But by far, they are definitely the most active in embryonic stem cells, placental cells, and cancer cells. And we think this has to do with the epigenetic environment of those cells.
And I think you're raising a great point, Vincent. And so, you could pick many systems maybe to sort of functionally dissect the contribution of these sites. One of the reasons we picked these STAT1 binding sites and thought about interferon gamma signaling or regulation is because there are clean phenotypes we can test. We know what an interferon-stimulated response looks like.
Right.
Whereas for some of the developmental stuff that Ed hinted at, that's a lot harder to say, well, how do we kind of disrupt this or experimentally manipulate it and then look at a developmental outcome in sort of the context of a whole organism. And so this is, I think, where the immune system and sort of a lot of the selective pressure that's placed on it by microbes and pathogens and so forth kind of gives us a nice foothold to functionally dissect this.
Hey, Nels, it would be cool to look at those 2 pigeon genes that your colleague has identified, see if they're within transposable elements.
Yeah, good idea. Now we're cross-pollinating between podcasts.
Yeah, that one hasn't come out yet, but
yeah.
I have a new respect for pigeons. All right, so next, Ed, you focused on a specific copy of the MER41 upstream of a very specific gene. Tell us why you did that and what you found.
Yeah, so that all that analysis is that's kind of the state of the field. So there are several papers that have done that kind of thing in different systems, as we just discussed. But now the goal was to take advantage of CRISPR, which had just come out at the time, or which had just become widely available at the time when we started the study, and just do the experiment of cutting out the viral element and seeing if it had any effect. And so, as Nels was saying, the innate immune system is wonderful because it's so well studied, and we can do a lot of these tests in vitro. We don't have to make a mutant, organism, which is a lot of work. So basically, what we started off with was we took those MER41 elements that were bound by STAT1, and then I just basically made a list of those elements and their closest gene. And the 3 of us sort of sat down and just pored over that list and looked for genes that may have had some well-known immune function. And it wasn't very hard. Many of these elements are near very interesting genes. But the most interesting to us at the time was a gene called AIM2, which is short for absent and melanoma 2. And we found this one MER41 viral element just about 200 base pairs upstream of AIM2. And it displays all the hallmarks of an interferon inducible enhancer. And so this was a very interesting element for us.
And what does AIM do? AIM2?
Yeah, and AIM2. So AIM2, this is
prevents
a double-stranded DNA. Yeah, so it was first discovered in a cDNA subtraction screen between melanoma cells and matched healthy cells. And this gene was a gene known to, or the second gene to be identified as absent in the melanoma cells. And so I guess the inference from that is that this gene is basically can act as a tumor suppressor. But then later on, about a little less than 10 years ago, it was sort of rediscovered as a double-stranded DNA sensor, important for innate immunity against a variety of different pathogens, including both bacteria and several double-stranded DNA viruses. So basically, this gene patrols the cytoplasm, and if any double-stranded DNA comes in the cytoplasm, which should normally not be the case, AIM2 will basically coat this double-stranded DNA and start assembling a complex known as the inflammasome. And this is a large complex that includes genes picard and caspase. And basically what that does is it does a mass cleavage of caspase-1. And then that cleavage of caspase-1 activates it and initiates an inflammatory response, including pyroptosis and also cleavage of cytokines such as interleukin-1 beta.
And pyroptosis is a form of cell death, right?
Yeah, it's a violent form of cell death. So instead of going quietly, it releases, a flood of cytokines to warn other cells that an infection is underway.
Burning down the cell. Yes.
Burning down the house. Burning down the house. Isn't that a song?
It's a song.
It's a good song.
Yes.
So you use CRISPR to do something to this.
Something to this element. So AIM2, the full name of this gene actually is Interferon gamma inducible absent in melanoma 2. So it's been it's been known for quite some time that this gene is normally not on in cells at all. And you have to, and this is in human cells, I might add. But you have to add interferon gamma, and to some extent interferon alpha can induce this more weakly to get AIM2 expression. So having that, and then knowing that there was a MER41 interferon inducible potential enhancer right next to it, really, made it tempting to delete that element and to see what would happen. So that's what we did. We did this in HeLa cells, and this was, our first time using CRISPR. Basically what we did is when we cut this out, we saw that this gene AIM2 was no longer able to be induced by interferon gamma. It was basically dead.
It's nice. Now, how long did it take you from the time you designed the oligos to getting the cells growing? How long was that, roughly?
I would say to the first result, it was actually pretty quick. I would say, maybe 3 months. So you design the oligos. One of the limiting steps is if you want to get a homozygous or a clone, like a clonal mutation, you have to do, you got to seed these at a single cell wells, and then grow those up, and then, genotype a couple of these and test them. So that's what we did, and that's what kind of takes the most time. The interesting snag here is that we were using HeLa cells, and initially we were using these just because they're really easy to transfect, so that kind of made CRISPR easier, and they're also easier to grow up from single clones. But we didn't expect is that this locus AIM2 is actually present in 4 copies in the HeLa genome instead of 2 copies of normal ones. And that made it really hard to get a complete homozygous deletion. So we were able to get, 3 out of 4 pretty constantly. But getting 4 out of 4, that actually took, I would say about a year and screening of many thousands of clones. And,
so is this is that because the copies are in different chromosomes that are repeated, or they're tandem copies?
No, it's just a probability of making a deletion with CRISPR. So, using CRISPR, and if you're just trying to make, say, a frameshift mutation in a protein, you're just using one guide RNA to target an exon, and you're just trying to get one mutation. But when you're making a deletion, which is what we had to do because MER41 is a repetitive element, so we had designed our deletion guide RNAs to the edges where they sort of overlap both host sequence and TE sequence. Making a deletion requires, both of those cuts kind of happening at the same time and the cell repairing it through non-homologous end joining in a way that basically excises the repeat. And so that's the ideal case, but that only happens maybe 20% of the time. And the rest of the time you get just little indels at both pairs, but not an actual deletion of the element, or you get other surprises like an inversion and duplications of the element. So it's still kind of a messy, it's not quite as clean as they make it out to be. So there are a lot of surprises that we ran into. But in the end, we were able to get a homozygous clone. So that was a win, I think. The champagne really got me.
Right.
That's right.
Yeah, on paper it sounds easy, but in practice it's never easy.
Yeah, CRISPR kind of rolls off the tongue, but when you actually are kind of in the trenches, I watched Ed spend a good several months chasing after this and really making sure that he had hit all 4 copies in a precise way.
I'm asking because we are trying to make a whole genome CRISPR knockout library of HeLa cells, right?
Mm-hmm.
And because we'd like to identify a receptor for a virus that way, and
cool.
It's probably going to be hard.
Yeah, so one of the advantages you have is, so I think people generally think of HeLa cells as these kind of crazy mixed up cancer cells with crazy, aneuploidy. Jay Shinduri's lab up at Genome Sciences at University of Washington has spent some time getting a haplotype resolved genome using sort of advanced approaches to doing this, and have published a really nice, it was sort of controversial actually, Jay's group and then a group in Germany, when they published the HeLa genome, ran into some ethical considerations for good reasons.
Right.
But it is out now. And so that allows you to know, and for Ed to know, for example, when he was doing his experiments on AIM2 or near AIM2, that we had 4 copies to target because there are these high resolution annotations of HeLa. So that's worth paying attention to.
Nice. Good to know. So, Ed, you actually did a virus experiment here.
Yes, being in Nels' lab.
Is that his idea?
I mean, it's a combined idea, I'd say.
Okay.
Yeah, so I can go into that.
Yep.
Basically, so right after deleting this element, we were able to show, based on RNA and protein levels, that AIM2 was no longer induced. But I mean, of course, the real question is what happens when you actually introduce a pathogen and you don't necessarily, cheat by pre-treating with interferon in the dish. Right. So that's what we did. So in this experiment, I actually didn't pre-treat with interferon. I just threw the virus on them. And basically what we found is that what we were assaying was caspase-1 cleavage. So this is kind of immediately downstream of activation of AIM2. And basically what we found is that if we looked for the cleaved product of caspase-1 after infection with vaccinia, we very clearly see cleaved product with in normal cells, saying that, so in normal HeLa cells, this pathway is functional. But in these mutant cells where that viral element is now gone, what we see is basically very little cleaved caspase-1. It's basically saying that this pathway has been broken, in these mutant cells through the deletion of this viral element, not the gene itself.
Right. Yeah, I think that was a really nice experiment. You have this lovely reagent now. We have paired cells that.
I mean, it was fortuitous that this gene was first studied with vaccinia, and that we're in a vaccinia lab that just happens to have a ton of vaccinia stock. So it wasn't wasn't too much work to get this experiment going.
Yeah.
And so just to mention it, I think it was Kate Fitzgerald's lab that connected AIM2 as a DNA sensor using vaccinia virus, and I think they used a couple of other pathogens with double-strand DNA.
Mm-hmm. Now, I noticed you also found MER41 close to a couple of other interferon-stimulated genes, and that's really interesting. One of them is ApoL1, and that's involved in trypanosome clearance, right?
Yeah, and I think there's some evidence that it's involved in HIV restriction as well.
Interesting.
Yeah.
And I know the trypanosome connection because Jane Raper, who is here in New York City at Hunter College, she works on this, and is actually involved in making transgenic cows with the human gene to try and make them resistant. So this would be for Africa, Dickson.
I hear you.
And because here we don't need to do that, obviously, because we don't.
No.
As far as I know, Dickson, we don't have trypanosomes here in the U.S., right?
We do have trypanosomes here in the U.S.
That infect people?
Yes, they do.
Is that right? Where are they? In your freezer?
They're in the South.
Are they spread through cows?
No, they're not.
Which species are they?
We have T. cruzi.
In the south of the U.S.?
That's correct. And then we also have T. equiperdum, which is a sexually transmitted horse parasite. Doesn't require a tsetse fly.
No, it's when horses have sex with horses, right?
That's exactly right. When they're horsing around, so to speak.
All right. Okay. Anyway, she's making these transgenic cows for use in Africa.
Right.
I don't know if they would actually.
Why would they ever want to introduce cows into Africa? That's the biggest question I've always had about that whole scheme. Is that they've got a lot of ungulates there already that they could easily, just
well,
eat.
Dickson, we're going to have her on TWiV in at some point. Well, you can ask her.
I was at ILRAD once when they were originally opening up way back when. That's the International Laboratory for Research on Animal Diseases in Nairobi. And I kept asking, why do you guys want to introduce cows into East Africa when you've already got all these herbivores at your doorstep?
Maybe they want to eat them.
Maybe. He says, have you ever tried to eat an eland? I said, no. This is tough as nails.
That's a good name for a podcast. Have you ever tried to eat an eland? I don't know when we would use it, but so Ed, you checked each one of these by deleting out the element, right?
Yes.
It was APOL1, IFI6, and SECT-M1, right?
Uh-huh.
And same result.
Similar. So these were genes that, again, we chose them on purpose because they all had, previously reported roles in immunity.
Yeah.
And so we deleted the MER41 elements. And each of these cases, the MER41, so these are different from the AIM2 case in two ways. First, they're all a little bit further away, so they're not, right at that promoter element. And second, they weren't the only STAT1 binding site. So if you look at the figure, they're actually one of two or one of four. And this is speaking to in the gene regulatory field. We think that a lot of regulation is actually controlled by redundant enhancers or shadow enhancers, so to speak, not just a single one. So in any case, when we deleted these, we didn't see that full on, total inhibition of activity. We still see that, for instance, in APOL1, when we add interferon gamma in the mutant cells where we deleted the viral element, we actually still do get we still get induction of APOL1. It's nowhere near as high as in the mutant cells, but there still is some expression. So this is saying that in these other genes, the viral element may not be responsible for 100% of the expression, but it does seem to contribute at least, 50% or more.
I have two questions. One,
sure.
Were these as hard to make? Were there multiple copies of these? Did it take a year each?
No, actually. This set of experiments actually started with the other ones because I was just getting so impatient about getting that quadruple homozygous clone. I looked for other genes that, using Jay Shinduri's paper as a reference, I looked for genes that were not present in 40. So these were actually present in 2 or 3, which was enormously easier to make. So these experiments, and by this time I had already done this 1,000 times. So these ones were actually pretty easy to make. It only took about a month to make these guys.
So you mentioned early in the paper that this particular endogenous element is quite old. I think 40, 60 million years old, right?
Yeah.
So it went into ancestors of humans. So can you trace it in other species as well?
Yeah, we can. So this element in particular, MER41, most copies are pretty much shared between human and marmoset. So basically all anthropoid primates pretty much have this element pretty much in the same place. There's going to be some differences, but it's hard to tell whether it's due to genome annotations and that kind of thing. But yeah, it's an oldish element. But I think what surprised us is when we were looking at the evolution of this element, and as is often the case with endogenous retroviruses, we actually see relatives of this virus in other mammalian lineages. So that includes lemurs, fruit bats, carnivores, cows. And
cows.
What was really interesting was that when we looked at these other lineages, even though the MER41 was traceable based on sequence homology, the insertions in those other genomes were completely different than the insertions in the primate genome. So, while cow had a MER41, they don't have a MER41 in front of AIM2 or these other genes. Their MER41s are in completely different places. And so what that says is that, maybe there was a family of MER41 viruses, and these kind of independently invaded different mammalian lineages, all in this 40 to 60 million year ago window.
Right.
Sorry, go ahead.
I was just going to ask, now that we have the Neanderthal genome, and of course we have our own, how do they compare with regards to the results you've got?
So with MER40, I haven't looked, personally. But with MER41, I wouldn't expect them to be too different between Neanderthal and human, since again, I think even between human and rhesus or marmoset, our MER41s are pretty much the same. That being said, there may be some more recent transposable elements, including ALUs or lines or HERV-Ks, that are, polymorphic between Neanderthals and humans. So, I mean, certainly possible that they might, some of these elements may be responsible for some differences. Yeah.
So let's say this MER41 went into a animal 40 million years ago. How long would it take for selection to have that element in all of the species globally? Roughly, I know you can't tell me exactly, but how long do you think it would take? It depends on the reproductive. So let's say, how often do mice reproduce, Dickson?
Pretty rapidly, every 6 weeks.
Every 6 weeks. So let's take a mouse to make it easier. How long would it take? Would it take a year, 10,000 years, 100,000 years? You guys ever think about that?
Yeah, we think about it. I'll step in here and I'll let Elde answer as well. But we think about this thing a lot. And so I think what really matters in these cases are, what's the sort of strength of the selection acting on it.
Right.
And so if that is a very strong immune scenario where you have an ancient population that's getting picked off by some suite of microbes, then this could move pretty fast. And whereas if it's something that's sort of fine-tuning evolution where maybe, you have one developmental pathway that sort of happens 5 seconds or 20 seconds earlier, that would be a much more subtle scenario and would take a lot longer to happen.
So let's say it's the strong selection. They're getting picked off.
Yep. So if they're getting picked off, I mean, this can happen over the course of just several generations, I would say. Maybe, 10 to 20 generations, perhaps, if it's a strong enough pathogen. And so it's a bit hard to really put those numbers. I know you want concrete numbers. We do too. But as we're kind of inferring these things, it's pretty tricky because we're talking about, stuff that happened way before our medical records, before and all of this kind of stuff.
Nels, I'm not letting you go until you give me a concrete number. Let's say you took...
Don't come back until you...
Let's say a gene went into a species, let's say humans, today, that was incredibly beneficial for some incredibly strong selection. How long would it take to be in all human populations?
Yeah, so here's, I think, a great example of that. And I'm going to skirt the question, but the great example.
Thank you, Senator.
Yeah, that's right. Is at least I'm admitting it. Is CCR5 deletions with the HIV as a receptor to get into the cell. And so if you look and again, it's really tricky because we now are kind of putting our thumbs on the scale of the evolutionary process with medical interventions, all the way to cures. But if you look in some of these populations, specifically in Africa, who don't enjoy some of the medical interventions that are happening in the West, you can start to track the rise of some of these alleles, and they are increasing in frequency. And so, it gets more complicated than when you extend that question for it to take over all, individuals in a species around the world, where you have now variable exposure to pathogens, different lifestyles, different environmental factors. And so this is, population geneticists are on the case. It's a really interesting question that and we're interested in. A lot of people are. It's really hard. It's a complex, messy world out there.
And you could look at something like sickle cell trait,
right?
For one that's already gone through this transition, which presumably took some length of time that did not completely overlap with the human migration out of Africa, because obviously the European populations didn't end up with it, but a lot of African populations did. And maybe you could narrow down how long something that had not a huge selective pressure, but a pretty significant selective pressure in its favor took to diffuse through the population.
Right. Yeah, and I think people are doing that. And that's one of the things that I think is really striking about, so the approach that we often take in these scenarios is to ask, what are the fixed differences in extant primate species? And so sort of one of the implications of that is that these things really have swept all the way through the remaining populations that are out there. But in a lot of cases, the geography of these species is a lot more limited than our own these days. And so you also, I think, you have to take that into account as you're thinking about the history of our species before we covered the entire world, that how long it might take for something to fix today is very different than it might have back when we were just kind of getting started.
Plus, I often think about this too, even though I don't have any reason for doing it other than I'm very, very curious, nor do I have the tools to really investigate it. But I'm thinking about the eradication of Neanderthals. I keep coming back to them because it happened so rapidly.
Yeah.
All right. I mean, it happened within a matter of 10 or 20,000 years after humans that were immigrating out of Africa for the second time brought with them. What did they bring with them? And I think they brought a lot of infectious diseases that to a Neanderthal would have been lethal. To a human, it would have not been lethal because we were living with them for a much longer period of time. Yeah. I've also heard the statement that Neanderthals' MHC region is a lot more restricted than Homo sapiens. So those are other things besides just immune factors that you guys have identified that could be the reason why you get survival too, right? Or you get a non-susceptibility to an organism, like, for instance, with plague when it swept through Europe, or smallpox, the same thing.
Yeah, I agree. We see these kind of echoes in those cases of smallpox or plague as you mismatch this with populations. I mean, and there it's also, I think also there's another layer of interesting sort of genetic history here as well, which is as people are mapping the introgressions of Neanderthals that remain in our genomes, and the idea, Josh Akey just had a really interesting paper in the last few weeks claiming that it might even be that some of what's left of the Neanderthal heritage in our genomes actually is there because of it's providing immune functions in some ways. And so that'll be, sort of coming at it from that population genetics level is cool hypothesis generation for then us to sort of pick up some of these things and ask from a functional standpoint, can we actually see differences that could impact basically the survival of our own species given some of the pathogenic challenges that we faced.
Right.
Nels, do you have any sense for what selected for the deletion CCR5 delta 32 in the HIV co-receptor?
Yeah, I think that one's also kind of controversial, right? And this is one of the tricky parts about this, I think, of any evolutionary sort of approach, is that we're trying to kind of come up with an explanation where kind of almost by definition we have to be covering vast timescales and vast geographies, and so it's pretty. You have to be pretty humble in your approach to saying that we really have this thing nailed. Kind of take the data as it comes and weigh it, whether it's some contribution of the plague versus the ancient retroviruses or some combination. But we've also seen cases like for CCR5, for example, where there are other genes. Lactoferrin, this iron-binding protein, is right in the neighborhood. And we think some of its population genetics has been influenced by bacterial pathogens over time. And so and then once you kind of crash all this stuff together into the messy complexity of real life, that's pretty tricky to then pull that apart with sort of a reductionist approach. And so
it is, but it's fun.
It's really fun.
It's such cool stuff. All right, so I think we can move to the end here. I want you, Ed, to tell us about this idea that we actually stole something that a retrovirus had first figured out.
Yeah, sure. So what we're thinking with MER41, as I mentioned earlier, is that when we look at MER41 copies, that they actually have that motif for STAT1 binding sites. And when we looked at these MER41 relatives that were in other lineages, they were close to MER41, but pretty divergent. But what was really surprising to us was that they also had those STAT1 binding sites in the same place. So there seemed to be conservation of those binding sites in the ancestral infectious retroviral genome. So we're kind of thinking that before, our analysis suggests that if we were to reconstruct the sequence of these viruses before they, when they first endogenized in the genome, suggests that all these viruses had those STAT1 binding sites upon integration. So, so to me, a non-virologist, from the start, thinking about why would a virus want to be regulated by the very pathway that sort of is made to kill it. But then as I did more reading, I started to realize that a lot of viruses actually have these similar kinds of regulatory properties or evolutionary strategies, if you will. And HIV is a really good example where there's a NF-kappa B and IRF7 binding site that's essential for its regulation. And, there's other both retroviruses and other viruses seem to commonly employ this strategy.
It's a great way to time. It's a great way to time a viral life cycle.
Yeah.
Exactly. Yeah. So especially with retroviruses that want to, that perhaps maybe want to be latent or, not do anything while its host cell is, self-renewing or replicating, and then only turn on when there's a secondary infection or stress. That seems to be actually a very viable evolutionary strategy. Again, we saw not just MER41, we saw 20 other ERV families that had this regulatory property. We're seeing some, totally unrelated ERVs in mouse. This RLTR30 also has 2 STAT1 binding sites, but very clearly independently evolved. So we think that the presence of these inducible binding sites might actually be a pervasive or common strategy with retroviruses. And if that were the case, then I think as these retroviruses continuously invade and integrate into our genomes, they sort of inadvertently also distribute these binding sites, which they first evolved, but now they're distributing these into our genomes, which we've shown that we can then co-opt and recycle these sites into our switches for our own innate immune responses. So it's kind of taking that same regulatory sequence, but sort of switching sides.
So what started as a viral strategy for exploiting the host immune system turned into a host strategy for exploiting the virus's exploitation of the immune system.
Yeah.
That's what we speculate. Yeah, I mean,
it's kind of a meta immunity.
Well, the other thing is that the pathogen has become a symbiont in one sense because it wants to keep its host alive so its own DNA can replicate.
I mean, we domesticated the retrovirus.
There you go. Now that's anthropomorphizing a little bit.
We let you because you know the dangers of it, right?
I do.
Yes.
So Ed, before
trained professional on closed course.
Do not try this at home.
That's great. Trained professionals, exactly.
On a closed track.
On a closed course. I love it. So, Ed, what you're saying is before mammals were infected with these retroviruses, they did not have these binding sites in their genomes. They somehow regulated these pathways in other ways, and then they acquired these, and maybe it worked better, so they stayed.
Yeah, so I think so that's a really good question. So transposable elements, including ERVs, have been associated with this idea of rewiring regulatory networks or even wiring new regulatory networks. In this case, the STAT1 interferon response is quite ancient. I think it's present in all vertebrates. It's present in fish. So and this particular ERV, of course, is much more recent than that. So in this case, I think the initial interferon regulatory network, did evolve as sort of a normal host network. But I think it's just these retroviral elements sort of evolved later to hijack it and now can contribute to more recent or lineage-specific evolution. But certainly this response did exist beforehand, and I think these ERVs are just kind of promoting rapid evolution and turnover of that network, sort of accelerating, pushing the pedal of evolution, I think, in that sense.
One of my favorite places to go to, of course, isn't too far from you guys, to Vernal, to sit as a spectator and watch people mine the bones of old, like Stegosaurus and Brontosaurus and Tyrannosaurus Rex, and to think that they all became extinct at around 65 million years ago, and then there was nothing left except mammals after that, so the retroviruses went crazy in mammals because there was nothing else to go crazy in. I guess fish too, but not as many, and obviously the mammals just exploded after that, right? So this exploded with them.
That's right. And at some point, I think this could be a nice cross-pollination for TWiV, where there's a great journalist here, Brian Switek, who does covers a lot of the recent excavations that are continuing to go on. And I think he had a great line recently that says, "We have not yet reached peak dinosaur," right? Meaning there's still so much to be discovered. And so, to me, that's just really exciting to think about.
And actually, just if anybody's looking to hire a great journalist, Brian was just let go by National Geographic. His blog, which was a really, really good dinosaur science blog, they've decided not to continue it.
Not too bad.
Yeah, good point, Alan. I think that's National Geographic's loss, and hopefully somewhere else will take advantage of this great opportunity.
Yeah.
Hey, now that just brings me an idea, which is totally unrelated, but doesn't matter.
Yeah.
Janet Iwasa.
Yeah.
Can you get her on TWiV or something?
Great idea. Vincent's talking about one of my colleagues here in the biochemistry department, Janet Iwasa, who's a great cell biologist, and then she became a molecular animationist. She started taking courses like using Pixar level kind of graphics and animation. She's here at Utah and a great scientist and collaborator to team up with to do really sophisticated animations and representations. We've done some stuff with her in the past. Thanks for the idea. We'll grab her.
Write it down maybe when I'm there or something or before. That'd be fun.
Absolutely.
I've wanted to do that for a while. Many people have.
I have a new series called Walking with Viruses.
Walking with Viruses. It's a great idea.
Yeah. And Janet's done a bunch of HIV replication cycle animations.
Yeah.
Really stunning.
People are always telling me, writing, you should get her on, and I'd like. Now I just connected you two together at Utah, so you're the facilitator, Nels. So now, guys, we have a great example of co-option of a retroviral element. We have the placenta syncytium co-option. Are there any other great examples like these two that you guys know of? I mean, on TWiV a long time ago, we talked about this long non-coding RNA from an endogenous retrovirus, which is supposed to maintain embryonic stem cell pluripotency. But I've heard,
I was going to bring that one up.
I've heard people that not so happy with that. So I don't know what you guys think.
Yeah, I agree. I mean, I think it's a clear. So in these cases, I think they're finding a clear phenotype. But again, it's in a dish, and whether increased proliferation or self-renewal of an embryonic stem cell is actually something that matters, to a developing organism. I think that's still an open question. So, again, these were, those kind of issues were the part of our rationale for studying the innate immune system in our study.
Got it.
Yeah, and I'll draw another Utah link here. So there's a fellow, another postdoc, one of Ed's colleagues, called Ed Groh, who just joined Brad Cairns' lab not too long ago. He was a grad student with Joanna Wysocka doing some research related to this, where they've been looking at some of the endogenous retroviruses and as they're expressed in some of these stem cell scenarios. And so I agree with Ed that I think it's a little bit tougher to put your thumb on it, but I think there's something real there, and this is being actively engaged here at Utah by Ed and other labs as well.
All right, is there anything we missed, Ned? Nels and Ed. I made a hybrid of you two, Ed.
I'm glad you didn't ask me that question.
I could say that and address you both at once, right?
There you go. Very efficient. I'll add one last point, maybe sort of something, just sort of a take-home message, and then I'll let Ed chime in if he wants to. But so, going back to AIM2 specifically, and the idea that, we're talking about this MER41 element being old, and it is old, 40 million years, but it's not super old. And so, we often think of the mouse as a great model system for vertebrate or mammalian biology, and it is in many ways. But if you were relying on the mouse to understand the regulation of AIM2, you would be lost because MER41 is a primate-specific element. It's absent in mouse, and in fact, AIM2 is not an interferon-regulated gene in mouse. It's there. The family is diversified, and now there's a post-translational mechanism that keeps AIM2 in check. It has to be degraded for AIM2 to become active. And so, I think this is one of the important things about thinking about the evolution of the immune system in particular, is that these things are so kind of volatile and specific to populations that in order to really understand the biology underlying this, that you have to kind of keep an eye on what rapid evolution can mean and how it can kind of scramble the regulation in different systems and sort of make model systems in some cases, if anything, deceiving instead of helpful.
Ed, any thoughts?
No, I think that's it for me.
What do you do? You're going to stick around there for a little bit longer, I presume. Are you continuing this line? Are you doing something different?
Yeah, I think there's a lot. I know this study, raises a lot of, or opens a lot of doors to different projects. I think we're still thinking about what to do next, but I think I have one project left in me here, at least. But I think one of the things we want to look at is let's do this in mouse and see if there's is this given how many ERVs there are in the genome. To some, it's not a surprise that some of these elements may be co-opted, and I think the next question from here is, well, are these just a few are there just a few cases of these in the genome, or is this just a tip of the iceberg? And do we actually see a pervasive co-option, not only in humans or HeLa cells, but also like in mouse, other species, other kinds of cells? So I think we're trying to just broaden our view and see how pervasive this mechanism really is in evolution.
Right.
Yeah, and so I'd say stay tuned for the next chapter. Ed's already got some interesting stuff underway. He's being a little cagey, but that's okay. And also stay tuned if you're a faculty hiring committee, because this guy would be someone to definitely take a run at
He's nearing his gestation period.
That's right. Absolutely.
All right.
I just want to point out, I think it's cool that, and probably when you do transposable elements, you get to do this a lot, but that you get to cite Barbara McClintock paper from 1950 or Britton and Davidson from 1969. It's just really neat.
Yeah, these ideas are very old, but I think we really, right now is the golden age where the technologies have developed enough that we can actually address those questions functionally.
Yeah.
Mm-hmm.
Well, this episode of TWiV is also sponsored by the American Society for Virology, and I want to tell you about this year's annual meeting, which is going to be held in Blacksburg, Virginia, at Virginia Tech University from June 18th through the 22nd. You're not going to be there, are you, Nels?
Unfortunately, no. So I am going to be at the ASM microbe meeting.
Wonderful.
And so we'll connect there. Yeah, definitely. Unfortunately, I have strung together a couple other meetings right at the same time. So, but ASV is on my menu most years. Just
all right.
This one. Yep.
Local organizers for this ASV, XJ Meng and Zach Edelman, and their committee are arranging this meeting. They put together a great program along with, of course, the program committee, of which Kathy is in charge, and the president who does the morning the talks, and that's Grant McFadden, of course. The keynote address this year is by Dr. Stephen Russell about oncolytic viral therapy, 5 days of symposia, workshops, poster sessions, state-of-the-art lectures. You can come listen to Dr. Joe Handelsman talk about leading the White House Office of Science and Technology Policy. She used to be on TWIM.
She's awesome.
You can be part of a live TWiV audience, because at ASV we do not encourage dead audiences.
No.
You can enhance your presentation skills at the Career Development Workshop. And this year, ASV is hosting a special Zika virus oral workshop and poster sessions, and they have issued a call for late-breaking Zika abstracts with a deadline of May 16th. You have until May 20th to register and get early bird rates, save a little bit of money. Go to asv2016.cpe.vt.edu, or you can go to asv.org and you'll see a link to the meeting there. A little bit easier. Hope to see you all there. We'll be there. I will be there. Kathy will be there. Who else?
Rich Condit.
Rich Condit will be there.
I will probably not make it.
I won't be there, but a couple of folks from my lab will. A postdoc called Dustin Hanks is giving a talk, and grad student Della Fixon also giving a talk. And another grad student, Kelsey Rogers, I haven't heard yet if she's giving a talk, but she'll be there.
I actually got invited to host a lunch table discussion about science journalism careers, but I'm probably not going to be able to do that. I still haven't answered him yet.
Oh, okay. Tell him to give you a rain check.
For how much?
A bucket full.
Yeah.
Let's do a couple of email. You guys want to stick around?
Sure.
Nels and Ed?
We're here.
Sure. Yeah.
I know you're dying to go camping, but just a little bit longer. The first one's from Suzanne, who writes, on so. I hear every so in interviews on the radio and in podcasts. Some days they drive me nuts. But until you mentioned it, I didn't notice that you use the word as often as you do. I think it's because you're using it properly. So has become the new for a lot of people. They stick it on the beginning of a thought while they're gathering their thoughts. You use it to connect a following thought to a previous one the way it's meant to work. I suppose it's still usually an unnecessary word, and who am I to tell you what to change about yourself, but I wanted at least to say you're not abusing the word. Mostly, you use it a lot when you seem to be excited about something, which is cool. So I complained that I use so too much. I hate it. I don't like it. And for the reason Suzanne points out, that you're gathering your thoughts, right? But she seems to think it's okay. Thank you. You made my day, Suzanne. I really appreciate it. Alan, can you take the next one, please?
Sure. Ricardo writes, hello, TWiV friends. First of all, let me thank you for the enormous hours of fun and especially learning. The weather here is beautiful for a primavera, spring day. 13 degrees Celsius with no clouds in the sky and 40% humidity, 3 UV, wind from the east at 22 kilometers an hour with 0 dew point. Now for science. In volume 531, number 7593, Nature magazine raises an interesting question. The issue is about CRISPR and genome editing, but I notice particularly the term citizen science. I think it has been an arc on TWiV, an interesting curiosity. I remember a basement lab with wonderful microscopes, but a curiosity. So the question is, in a time when we can literally build a lab in anyone's backyard, is CRISPR or other similar technology, the technology that will bring the home lab out of the curiosity and to relevance for the world? Will that be a problem or an advancement? P.S. In my opinion, it might be an advancement, as we've seen in software and electronics, but we really need better education. And Ricardo is an associate professor of microbiology at Fernando Pessoa University in Portugal.
Yeah, so maybe Ed wants to weigh in on on how easy it is to do CRISPR in practice.
Yeah, I mean, my background is in computational biology, so I guess I'd consider myself a computer guy. And if I could do CRISPR, I think anyone could.
Well, it wasn't
I think
easy.
the question. I mean, but that's just time. That's not necessarily money or. But I think. So I think CRISPR could be in the hands of basement labs, but I think it's really what you do afterwards that kind of matters. I mean, I think cutting up the genes of cells willy-nilly, that's one thing, but I think an actual experiment needs to follow, and that can be tougher to do in a basement lab, but willing to be surprised.
I just don't know what I mean. Yeah, what is the phenotype? You can put GFP into a cell in your garage, and that's cool. You can see them glowing, right?
Yeah, I think that's what like the CRISPR kits are these days, right? It shows you can do it, and so I think maybe as an educational tool, it could be pretty cool. CRISPR for kids. But as far as like major scientific breakthroughs, I don't.
That's the name of a company. You ought to start it. It's CRISPR for Kids.
CRISPR for Kids.
I did see there's a new thing called the Bento Lab that I think was a Kickstarter. I don't know the details of it, but it's sort of this one all-in-one PCR machine / kind of other functions. And so
as the Japanese name suggests, it's a lab in a lunchbox.
That's right. Yeah, exactly. So there are these cool advances, and I mean, what fun to bring some of these molecular techniques to basements or backyards, or I'm all for citizen science. I think it's a great movement.
Yep. The next one is from our friend Igor here in New York City at the Global HIV Vaccine Enterprise, who points out the great 3D model of Zika virus from Visual Science, which was a pick on TWiV 380. So I guess Igor is a little bit behind in his listening. And Kathy, can you take Mauricio's, please?
Sure. He writes, Dear Twivers, I was extremely excited when I heard you were talking about a plant virus, but I got even more excited when I realized that you had found interesting, or hopefully fascinating, that some plant viruses have multipartite genomes and its implications with efficient cell infection. Some viruses are only bipartite, but some others, like cowpea chlorotic mottle virus, CCMV, or its cousin, brome mosaic virus, have 4 RNAs. RNAs 1 and 2 are packaged individually in different capsids, while RNAs 3 and the subgenomic RNA 4 are always co-packaged in a 1-to-1 mole ratio in a single virion. Furthermore, the subgenomic RNA 4 is not necessarily for infection, but is always co-packaged. As a side note, when I became an independent researcher, I wanted to really understand co-packaging of the subgenomic RNA 4. The fascinating thing is that in order to produce an infection, at least 3 virions containing the 4 RNAs have to localize within the same cell, or at least within neighboring cells. I find this fascinating. My PhD thesis focused on understanding the physical rules by which CCMV packages its genome. How small and how big can a single-strand RNA be in order to be packaged by CCMV capsid protein? How does in vitro packaging efficiency depend on the length of the packaged RNA? And how does the assembly mechanism depend on the length of the RNA? All of this work was done in vitro with purified capsid proteins from the virions and in vitro transcribed RNAs. The main points are that packaging efficiency is maximum at the length of the RNA 1, 3.2 kilobases, and that as you go shorter or longer, the efficiency decreases. The short RNAs are preferentially co-packaged, and this assembly mechanism is different from that of the long RNAs, around 3.2 kilobases. And when the RNAs are too long, they cannot then fit within a capsid. I won't tell you what happens when the RNA is too long. I will be mean, hence you will need to read at least one of the papers to figure out what happens. Hint, Cadena Neva et al. I hope you enjoy them. All the best, Mauricio. And he's in the HIV Dynamics and Replication program at the National Cancer Institute. Did he send you links, or we just have to go find Cadena Neva on our own?
Yeah, unfortunately, he did send the articles. I didn't put them in there. So
okay.
my bad. So we can't, but we'll do it. We'll work on it. We'll do it another time because this is cool stuff, which I had not been aware of, and it's really neat. You can do these things, and I think this whole packaging and separate genomes is a really cool thing that not many people pay a lot of attention to. So yeah, we got excited about it last time. That was pretty cool. So we'll have a look at those, Mauricio. We'll tell you next time.
Yeah.
All right. So Dickson.
Yes.
Can you read?
Last time I looked, I could.
The next one is from Marie.
I know. I'm going to say that, actually. Marie writes, but she doesn't say TWiVers or anything. I'm surprised. Marie writes, listening to a TWiV 376, I believe, you mentioned an outbreak of anencephaly along the Mexico-Texas border, and it not being linked to a known pathogen. I thought about these papers showing it being linked to mycotoxins. Fumonisin. Is that how it's pronounced?
Fumonisin?
Fumonisin, that's the one. That is produced by some species of Fusarium. I believe this might be the answer to the question that you were asking, and then she lists 2 papers, one by Morass et al., and the other one by Mismer et al., in which those 2 compounds are linked to these teratogenic effects.
Well, that's cool because we were wondering because I think it was what was the name of that virus? Border something virus. Anyone remember? It was a specific virus that they looked for, and it was not correlated with these anencephaly. But she thinks it's fumonisin, which is cool. Yeah.
So she concludes always listening for you to include plant virus.
Marie is a professor at South Dakota State University.
Right.
Who must study plant viruses.
Indeed.
And wants to hear more. We will try.
Right.
Yes.
We will try to do that.
Yeah.
The next one is from Suzanne again, who said, I was just following links from your show notes and realized I ought to thank you for them. All your podcasts have the most organized, useful, and easy-to-use show notes of any podcast I listen to professionally or individually produced, and I think I'm up to about 50 now. Thanks.
Wow.
So Suzanne is the same Suzanne, and she's made my day twice.
Yeah.
Yes.
Yeah.
I didn't know anyone. Yeah, right. You're right, Dickson. It's not about me.
Just a joke there.
I didn't know that anyone actually looked at the show notes, but I dutifully make them every week in case one person somewhere. I think Kathy uses them a little bit.
We use them a lot.
I use them too. Yeah.
So I enjoy doing it. Hey, Nels, you want to read an email?
Sure.
Anthony
writes, blood sharing in vampire bats has been studied. It's been shown that this apparent altruism involves help me out, someone.
Reciprocity.
Thank you. Reciprocity, not
go.
Selfish genes. And then he has a link to a paper from the Royal Society.
Yeah.
I hadn't heard about rabies being spread by blood sharing, and a quick Google search doesn't seem to show it. My understanding is that to infect the rabies virus needs to be transmitted by a bite. A trip through the GI winds up nowhere, the gastrointestinal tract. And even if rabies was spread by blood sharing, it wouldn't explain how North American bats, mostly insectivores, catch the disease. And on the subject of bats, in a recent TWiV, there's a good cross-pollinating connection here. The paper discussed states that all cell lines were maintained in a humidified 37 degrees C. My understanding is that bats, with the exception of breeding females, go into a daytime torpor with much higher temperatures during flight. Might similar temperature change in cell cultures be something to look at? So this is the TWiV episode where we were discussing the evolution of Ebola virus, and in particular the straw-colored bats, and they were using cell lines from various species. Anthony continues, also there was a mention of not being able to bring specimens to the U.S. Was this because of an oversight in obtaining the permits or because of some daunting aspect of the paperwork? I believe that there also was something about a lab in Africa inadvertently destroying specimens. In what country did this happen? Was the error egregious or unavoidable? Much work is going to be needed on African bats, and it will be great if other researchers can avoid hurdles. Thank you.
I think that the temperature is a good idea, right?
Yeah.
I agree, because that's, I think, a pretty dynamic scenario for...
It's going to be tough to hang the incubator upside down from the sun.
But not impossible.
But you could do it, Alan, because you like doing that sort of
Yes,
thing.
I like modifying lab equipment.
The importation, Nels, I think, was because they just can't bring them in the country, right?
Yeah, that's right. And so, we could connect Anthony through to Kartik or Sarah Sawyer to get at some of those details. But I think you're right, Vincent.
Yeah, I can send this on to them. And destroying specimens, well, unfortunately that happens, right?
Right.
Yeah.
I can imagine.
Right, but the solution to an importation problem like that is ideally would be to set up collaborations and labs in Africa where you could do this. Which would pay a lot of dividends in the long run, I think.
Yeah.
Agreed.
All right, let's do two more short ones here. Alan, can you take the next one?
Sure. Robert writes, "Good afternoon, TWiV folks. Apropos the discussions of Kevin Folta and Monsanto, this appeared this week from WBEZ, an NPR affiliate in Chicago, and links a news story quoting from that. A WBEZ investigation has found a University of Illinois professor was given more than $57,000 over less than 2 years from genetically modified organism maker Monsanto to travel, write, and speak about genetically modified organisms, including lobbying federal officials to halt further regulation on GMO products." Robert continues, "I also note that Whole Foods is launching a big GMO Your Right to Know campaign. I find it interesting that in their GMO frequently asked questions page, that missing among the list of questions is, why should I avoid GMOs?" Yes,
oh man, this is really bad. That this guy.
This is a really, really stupid move. This is not Kevin Folta. This is a different person, but just very clearly over the line. Took money directly from Monsanto and appears to have channeled it through an account that I assume this was the intent. It had the effect of partially concealing it. So it smacks of a secret payment. It's just wrong all over.
Thank you, Bob. All right, Kathy, can you take the last one, please?
Sure. Patrick writes, "Hi, TWiV. I'm presenting the Levasseur et al. Mimivir paper in our lab's journal club and came across the original PLOS One paper describing the Zamalon virophage. In the methods section, they described that it was named Zamalon, the neighbor, in Arabic. Well, that's all good. The real reason I'm writing is to tell you about the weather, which is 75 degrees Fahrenheit and sunny in Berkeley. Thanks as always, and can't wait for the Carl Zimmer episode. Patrick." Oh, that's great. I was just thinking about that today, the origin of that word.
Yep.
Oh, I know why, because we have something coming up about the second page of Google. So thanks, Patrick. That's great.
Patrick apparently has left the Malik lab and he's now working on innate responses to bacteria. Oh no, in the advanced lab at UC Berkeley.
Yep, I know Patrick well. He and I overlapped in Hermit's lab.
Yeah.
He's a great guy.
He's a frequent writer to TWiV.
Yep, great scientist. Keep an eye on this guy too.
All right,
nice.
I will. Okay, let's go to some picks of the week.
Yep.
Dickson, do you have a pick?
I sent it to you twice, in fact. Yes, I do.
I'm just asking on the show, like...
Oh, yes.
No, but so, Vincent, what you have to say is, Dickson, what is your pick?
Exactly.
Why? Why do I have to
Kathy.
do that? Why
That's
can't
That's the way you ask everybody else. You don't ask me that.
Why
You say,
can't-
Oh, by the way, Dickson, excuse me, I didn't mean to wake you.
Why can't he...
Why can't he?
Forgot now what I was going to say.
It doesn't matter.
Just answer me instead of telling me how many times.
have.
Is this thing here Realistic Dinosaur? Is that your
Yeah,
pick?
that's the one. If you play it.
You know why I
it's
do
good.
that, Kathy? Because sometimes in the old days, he used to say, no, I don't really have anything today.
That's true.
Which really burned me up.
Well, I
It's so
didn't.
bloody easy to have a pick.
Well,
Yeah.
there's my pick. So, have you seen this?
I went the whole episode.
Stop picking on Dickson.
What is your pick, Dickson?
It's that video.
Yeah, but they don't know what it is if you say that.
Well, it's a
He's
fake.
pointing.
It's a
Radio
velociraptor.
audience, Dickson.
Can't they tell what I'm doing?
I just pasted it in the show notes now.
Okay.
Everyone can.
So
go see.
it's a replica. It's a dinosaur suit that this guy has constructed that looks remarkably like a velociraptor.
He wears it?
He wears it, and he can scare the hell out of you. You watch what happens when this guy, in cahoots with some people down in a parking lot, call upstairs for someone to come down into the parking lot.
Oh my gosh, it's so realistic.
And it looks absolutely like a dinosaur.
He's in a parking space.
Great.
It is amazing.
All right, Dickson, I think this is a little childish.
No, it's called Jurassic Car Park.
It's from an Australian radio show called Hamish and Andy.
Hamish and Andy, that's right.
Alan, is that what happened to Hamish Young?
I guess so.
That's pretty funny.
It's a riot. But people could have died of heart attacks because this thing looks so real. I mean, you immediately believe that it's true.
If you saw this in your car park, Dickson, you'd probably freak. It's very realistic.
I mean, it even makes noises if you turn up the sound.
Although
hear this.
you should know scientifically that this could not happen.
Exactly. That's the point.
She's coming out the door. She's
coming out the door. Now watch what happens.
Holy crap!
She wants to leave, and this thing goes right into the door with her.
This is staged. She wasn't scared enough.
Oh, she was scared. She was
rationality would not kick in when you're looking at this thing.
That's correct.
That's the thing. Intellectually, you would know that this is not real, but that part of your brain would not be active as fast as the part that says, "Oh my God, it's a scary monster run."
Wow. One of the participants here said, "I knew it wasn't real, but I didn't believe myself." So it was really terrific.
Wow, that's pretty good, Dickson, I must say.
And I believe in aliens too, by the way.
Despite the silliness. Okay, thank you, Dickson. What do you have, Alan?
I have an actual science pick. This is a paper. It was there's a paper in Science, which is part of the pick, which just came out, and there's an animation that goes with it. The paper is closed access. The animation has been redone on a number of sites, including space.com, and you can go look at that. And that really is, I think, for most of us, is the cool part anyway. This is a simulation of the plasma flow on the surface of the sun.
Oh, right.
Which is a critically important subject for obvious reasons, but it's been really hard for physicists to come up with good models that show both the large-scale changes and the high-resolution small-scale changes that really replicate what's going on the surface. Yeah. And these folks seem to have come up with a model that does that. I don't really understand all the math behind it, but the animation is just really, really cool.
Yeah. It's cool.
Wow, that's nice.
Totally agree.
It's very nice. It's very cool.
Awesome.
I'm just looking at it here. Ooh. Yeah, Dickson and I. Okay, we'll stop.
It kind of soothes you. It's like a stare. You shouldn't stare directly at it.
Put it on your TV at night.
Dickson, you have to put some exposed film over your
exactly.
Okay.
Kathy, what do you have?
I have a pic that my older brother sent me. Evidently, he gets NSF press releases, and it's a press release that relates to a story in a journal, but the main headline is Potential Zika Virus Risk Estimated for 50 United States Cities. And they have this map, and they have the potential risk with different colors of dots. And so I learned a couple things about this. First of all, the lead author is from the National Center for Atmospheric Research, or NCAR, in Boulder, Colorado. And that's the place that my brother and I visited when I was at that same ASM meeting where Nels was on our TWiV. And so this is a really cool facility, and it was designed by IMPay, and it's up against the Flatiron Mountains, and so that's why I put that picture in the show notes.
That's cool.
The other thing I learned about is that the primary article itself that this press release refers to is in a journal called PLOS Currents, Outbreaks. And I didn't know there was such a thing. This is something that started in 2013.
Because just what we needed is more PLOS journals.
Right. Well, and there's PLOS Currents Outbreaks, and there's PLOS Currents other things. But at least they're open access. And so if people want to read the original article that this map came from.
And the map shows that Nels is going to be okay. Vincent has a little bit to worry about, and Rich is in deep trouble.
That's right.
So this image I showed to the OBGYN department last week when I talked about Zika, this is make that guy proud, the visual representation of results. Dickson, what's his name? He wrote that book. Edward Tufte. Edward Tufte. Because look at this, it's the U.S., and then they have a circle. The size of the circle tells you how many people come into that city, and then the top half is Aedes aegypti, the bottom half is Aedes albopictus. Oh, it is so cool. And you could just look at it and immediately get all the info.
It's a movie in a snapshot.
Beautifully done. Very nice.
That's right.
I like that very much. Thank you, Kathy. A nice picture there.
Yeah.
Mm-hmm.
We could do TWiV from there.
Yeah.
Oh,
Would be nice.
Definitely.
Nels, what do you have?
So, in the spirit of kind of podcasting and sharing podcasts and cross-pollinating, I picked a different podcast to highlight that I just came across yesterday. So this is called Don't Get Me Started. The hosts are two fellows called Anthony King and Will Hines. I think they're headquartered in Los Angeles. And the premise of the podcast is to get someone and they get some celebrities being in LA, and I think they're in the entertainment industry, but it's to get someone who is known for something, but to talk about something that they're not known for, or just sort of an interest that's something they're into. And so the reason I got onto this was because one of my grad school buddies, Matt Giorgiani, who's now a scientist in Sean Carroll's lab up at Madison, so an evolutionary geneticist, and his day job is to work on the evolution of snake venom in rattlesnakes and other critters like that. But he got pulled in, and I don't know what the connection was, onto this Don't Get Me Started podcast based on his sort of unusual and inordinate interest in William Howard Taft, the president.
Well, of course, those are natural connections. I don't know why anyone's confused.
I love the concept behind this podcast.
Yeah.
Yeah, it's a good idea.
That's a great idea. Take people who are involved in one field and talk to them about what they're interested in that's not what they primarily do.
Taft and snake venom. I like it.
Yeah, exactly. And I got to say, and this is the only episode I listened to, is my buddy Matt, but it really works. It was a lively conversation that went for about an hour. And
Cool.
Matt was really selling it. He knows a lot about Taft and really wove the history together with, FDR and Teddy Roosevelt, and then how Taft sort of is not as well known, but was right in the middle of all of this.
Sure.
And I won't do any spoilers, but so with Teddy Roosevelt, there's this famous the teddy bear that came from that.
Right.
With William Howard Taft, they tried to launch the Billy Possum with not as much success. But anyway, I highly recommend listening to this podcast and my buddy Matt's episode in particular. It was really fun.
Cool.
Politicians are good at playing dead, though.
There you go.
Nels, that reminds me of my pick on TWiV this week, which is Story Collider.
Hey.
Oh.
And I think I can't pick it twice, but StoryCollider.com is it's a podcast where Ben Lilly, who had me go to the Strand bookstore this week and chat about viruses, he has scientists go to clubs and do a comedy routine. Basically, they have to just talk about something in their career, 15 minutes, that's funny.
Oh, neat.
And yesterday I listened to George Church talking about something.
You ever listen to The Moth?
He didn't get. Yeah, that's what it's based on. Exactly.
Yeah.
Exactly.
Yeah, there's a great one with Mary Claire King. She tells an incredible story.
Yes. Yeah. In fact, Ben wants to do that here in the city. Ben Lilly wants to start a nightclub where it would be, they would have bars and you could buy drinks, but the people at the mics would be scientists. Not real comedians.
Oh, very good.
So that would be funny. You could go, Dickson. You could do that. Anyway, that's not my pick. My pick is, but I do think people would love Story Collider. It's pretty funny.
Yeah, well, and can I ask Vincent, how did it go at the Strand bookstore? Because you were mentioning it ahead of time.
It was just so much fun. Stuart Firestein, who's a neuroscientist down at the Morningside campus of Columbia, he and I sat in nice cushy chairs in front of an audience. People came, and it's recorded. They did video, so it'll be on YouTube in a few weeks, and I'll mention it around.
I'll give you a like.
I had a great time. You'll give me a like? Thank you.
Sure.
Jason, even though you don't like me.
It's not true at all.
Now,
I'm quite fond of you.
Nels, I have to tell you, back on TWiV 322, one of our listeners, Sandra, picked the Who Can You Trust Mary Claire King moth story that
cool.
I had sent her. So,
Oh, good. That's one of my favorites.
Hey, Ed, do you want to make a pick, or you don't have to?
Sure, I have a pick.
Wonderful.
So mine would be, it was a news article in Science. I don't think it's published yet, but the title is Why Watching Comb Jelly's Poop Has Stunned Evolutionary Biologists.
Yes.
I'm sure some of you guys have seen that one. And so, I'm not a marine biologist, of course, but apparently comb jellies are a very primitive animal that, scientists have thought for a long time had just one hole for eating and pooping. So they would eat, digest their food, and poop it out of their mouths. But apparently at Tenapalooza meeting recently, a scientist named William Brown used GFP tagged or red fluorescent protein tagged crustaceans
Interesting.
and zebrafish and had these comb jellies eat them. And then because they were glowing, because their food was glowing, he was able to observe the food actually not coming out just from the mouth, but also through little sphincter-like pores in the back of the comb jelly. Apparently, this is a very surprising result in the field, and a lot of people are excited about it. So, comb jellies are pretty cool if you find it.
They are.
I've heard that. I've heard of it. I don't know why I would, but it's not published yet. Oh, it is published.
Right.
Well, it's.
Nels and I just pasted it.
Yeah, there. It's published.
It was a tie.
Okay, sure.
Yeah.
there.
All right.
Nice. Very cool. Thank you, Ed. All right, I have two small picks. I'm sorry, they're a little self-serving, but one of them is an article I wrote on my blog, and I just want you to see this cool letter that someone on Twitter brought to my attention last week. It's a 1953 letter from
This is great.
Yeah.
from Harriet Efrusi Taylor to Jim Watson, where she says, "Top secret. Burnett swears from work in his lab that flu virus has principally, possibly exclusively RNA. Suspects same for polioviruses. Question mark, question mark." Handwritten letter from the Hotel Frankfurter Hof to Watson, wherever he was. I thought this was so cool.
Yeah.
And so I went and I did a little research about, and I wrote a blog post. But I want you to see this letter because it's a Cold Spring Harbor archive, and it's just so neat. And I even emailed it to David Baltimore, and he started telling me stories. He knew her, and she was interested in DNA transformation, right? And at the time, we all had said, yeah, DNA is the genetic code, and now all of a sudden these RNA viruses have these viruses that have RNA in them. And it was actually tobacco mosaic virus that showed the way as for RNA being genetic material. So that's really cool. But the other pick I wanted is an article someone wrote about my blog. What is her name here? She didn't sign it. Oh, she didn't sign the blog. Anyway, it's at a place called the ACI Scholarly Blog Index, which is always a problem. Places that collect blogs and call them
Tracy Hector?
Tracy Hector, thank you. She contacted me and did some interview by email. She wrote a nice little piece about the blog and asked me some neat questions about my past. And she said to me over email, it was one of the most exciting things I've ever read, and I felt badly. If this is the most exciting thing I've ever read. But anyway, she... I told her that Leo Laporte got me interested in podcasting. She even put a link to him in the article, which is very cool. So you should check that out. Well, you don't have to because it's about me, but I thought she did a nice job. That's why I want to send you there. And also the whole thing, the ACI Index of Blogs is pretty cool. They collect them in there and they write about them. So you might find some things that you like. So check that out. We have a couple of listener picks. Sal writes, "Hi, TWiV hosts. I thought you might find this article about Mendel, the father of genetics, fascinating. It briefly touches on different aspects of a scientist's scientific life. Did you know that Mendel sent 40 reprints of his results published in a low-impact journal to various botanists and biologists, including Darwin? I did not know it. I am sure that Dickson, the father of vertical farms, will like the article."
I'm sure I will.
Don't be so excited, Dickson. Here's someone.
I'm sure I will.
It's an article in the ASCB, and it's the lost paper of Gregor Mendel, the patron saint of low impact scientific publishing. It's cool, and they have a picture of the original article in German. Thank you. I'm a long-time listener, blog, Facebook, Twitter follower, and a second-time writer. Weather right now is not so Texan here in Dallas-Fort Worth, Texas, and by that I mean a mild temperature, 70% humidity, moderate wind speed, and isolated showers. In fact, my light sweater with a TWiV logo on it sufficed throughout the day today. Oh, this is just great. Love it.
Excellent.
Love it. All right, one more from Dennis. "Hi, Docs. Thanks for another wonderful TWiV 380. At the end, during the Pics section, I believe Rich Condit picked the YouTube physics girl who does wonderful explanation videos. Coincidentally, there was a recent article about a different physics girl prodigy at Harvard who was researching gravity and related fundamental phenomena. Her website is physicsgirl.com. She's definitely one to watch as her work breaks new barriers, as does she. So my pick for today is physicsgirl.com for Sabrina Gonzalez-Pastersky. Separately, the TWiV on Mage with Harris Wong was action and info-packed and answered a lot of questions. Best Dennis. So if you haven't heard our TWiV with Harris Wong, you should. He is just amazing. He was trained by George Church, all right? And this guy is doing whole genome mutagenesis in bacteria, and, you know, he wants to mutate the microbiome of mice, and he's got all these methods for doing it. Oh, it's so cool. The guy is amazing.
That's great.
And he's
take a look.
Take a look. You guys would like it.
Thanks.
And he is my colleague here at Columbia. He used to be on our floor, Dickson, right?
Yes.
Should I read the last one? Yeah, why not? Jacob writes, "Hi, while hearing your discussion about the second page of Google results reminded me of this XKCD comic, and you have to go look at it because it's pretty funny." You know, I just asked Kathy the other day whether she was looking on the second page of Google results, and she said never, right? Never do that.
No, we were talking about it on TWiV, and that's how this came up. Yeah.
I asked you if you ever looked at the second page.
Not very often.
When I type out vertical farm, I get up to page 15. I'm not kidding. That's how I find the new ones. And every now and then something pops out that's surprising.
Sometimes you
absolutely surprising.
Oh my gosh! Look, my listener pick is an interview in ACI about some guy you might know, and oh, I didn't know this. No, I can't pick it myself. Right?
You can.
Why not? You did the other two.
All right, I'm unpicking it and giving it all to Jacob.
There you go.
Will I have to edit out my talking about it?
No,
it's up to you.
No,
you can make it a listener pick.
You talk about the writer of it.
Yeah.
I would defer to Jacob. He's a molecular biologist in Sydney, Australia, where they're having scattered thunderstorms in 21 degrees Celsius. And that will do it for TWiV 382, which you can find at iTunes and also at microbe.tv/TWiV. And I would like to thank our guests today for joining us. Ed Chuong from the University of Utah. Thanks so much, Ed.
Yeah, thanks for having me.
Great work. Keep it up.
Thank you.
Nels Elde from University of Utah. You can find Nels at cellvolution.org and at Twitter, Earlybird, and also on TWiV. Thanks, Nels.
Yeah, hey, thanks for having me. This is super fun. We'll let Ed go. He's got a camping trip to get onto here.
Yeah, that's important.
But thanks for bringing in our paper. I really appreciate it. And if folks are interested in some of this kind of evolutionary crossover, check out Vincent and my podcast, TWiV.
You bet. Thanks, guys. Cool. Kathy Spindler is at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun.
Alan Dove is at turbidplaque.com. He's also on Twitter. Thank you, Alan.
Thanks. It's always a pleasure.
And here we have, right in my office, the father of the vertical farm, Dickson Despommier. Thank you, Dickson.
Welcome.
Always a pleasure, Dickson.
Yes, indeed. It was always a pleasure. It
always
still
a pleasure
is a pleasure
to have you.
It still is a pleasure.
I'm Vincent Racaniello. You can find me at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral. Okay, here are our titles. And listen carefully, Dickson.
I'm listening.
TWiV, I don't know how to say it, TWiVivo.
TWiVivo.
I'm actually reading them.
Repurposing selfish genetic elements. Yeah. Who's selfish now? Infiltrating the stat department. Uncle Irv has you covered. Fighting virus with virus. Everyone's a little bit viral. The 8%.
You can sing for that one. Everyone's a little bit viral. It's true.
Yes.
If Kathy agrees to sing that on the podcast, that's my vote. That is my vote.
You can make it an Easter egg if you want.
The 8% solution and PCR pretty clear result. How you want to do everyone's a little bit viral?
If we can get that Easter egg from Kathy.